So today we will be going to talk about northern blotting as a gene expression technique. So before going to talk about northern blotting, first have a look on what is blotting. Blotting is a process of detecting any macromolecules that we deal with like DNA, RNA or proteins. If it is a process of detecting DNA, we call it as southern blotting. And if it is a process of detecting RNA, we call it as northern blotting. And if it is a process of detecting protein, then it was given the name of western blotting. Because the names like with the directions, just like north, south, west, but actually it's nothing to do deal with the directions. The first type of plotting technique that was discovered in 1975 by E.M. Southern, on the name of the discoverer, it was named as Southern plotting. But later, in 1979, 4 years later, another technique was discovered that is known as Northern blotting. Because RNA and DNA are very closely linked. So earlier it was Southern blotting, that is why they named it as a northern blotting. Now, what is the process of northern blotting? It is very similar to that of southern blotting. In any blotting process, there are basically three major stages. The first stage is to prepare the target. The target in this case of northern blotting is the RNA. So, for an example, RNA is present inside the cell, in the cytosol of the cell, or inside the nucleus. So, what we can do here, we simply extract the content of the cell that should contain RNA, especially we do the process of this northern blotting. In case of messenger RNA or mRNA, and the content of mRNA is present inside the cytosol, so you take that RNA out, we extract that part of RNA, so we have an RNA mixture which is a whole RNA mixture of the cell. But again, like any other detection techniques of blotting, the idea is to find out a specific target of RNA which we know the sequence, by the way. So, how do you find a known sequence of RNA from a mixture of other RNAs? The only way to do that is the hybridization technique or nucleic acid hybridization. Because nucleics has a very important feature. For example, if I tell you the target RNA that we are looking for as a sequence such as AAUUAGGCUA. For example, this is the RNA sequence that we want to find out. Okay, we know that DNA and RNA both have a complementary feature. So if they find any complementary strand, they are going to attach to it. The complementary strand of this RNA will be UU AAUCCGAU. And if we think of this complementary strand as a DNA fragment, it could be TTAATCCGAT. So this is the complementary strand that we have to generate. So once we generate this strand, we can put that strand on a probe and that can bind to the target DNA. Now, if we attach this target strands along with some sort of radio label or radioactive molecule, or if we attach any sort of chemical or enzyme molecule which is giving us light, then after the reaction, we can accurately identify the position of the target RNA in the mixture of RNA while we separate it on the gel. Remember that is what we do in the northern blotting. Is the first step we do is to extract the RNA. Then we break those RNAs into smaller fragments because RNAs tend to present in complex secondary structure forms because they are single stranded, so they have the ability to pair with wherever they find a complementary region. So RNA never presents in a linear strand. They always present in a very complex structure and secondary structure form. We want linear structure of RNA and the fragments of linear structure of RNA. So for the process of nucleic acid probing or nucleic acid hybridization, then only we can find out the specific sequence of the RNA. But how can we break the complex structure of RNA into linear form? In that case, we need to use a denature in gel electrophoresis process, which will help us to resolve the secondary structure into linear RNA. Then we can separate that RNA using electrophoresis, which is an agarose gel electrophoresis process. Then we use the probe and then the process will be done. So this is whole stages will be known as the northern blotting. So first we do RNA extraction and we load this RNA onto the agarose gel. Let's say this is the agarose gel that we are talking about. This agarose gel is slightly different from agarose gel that we use in southern blotting. The agarose gel that we use in northern blotting contains formaldehyde. Why we use formaldehyde? Because this formaldehyde will help to resolve the secondary structure into the linear form of the RNA. And that is very, very important. Otherwise, the RNA will not be separated based on their length. The idea of using agarose gel is to separate nucleic acid contents based on their length because agarose is a polymer of small matrix and they will form small pores inside. So smaller length of RNA or DNA fragments can migrate further along the gel while the larger fragments or the longer fragments will migrate less in the gel. With the help of this, we can identify where the larger and smaller fragments of the colony are present. But if the RNA is in secondary structure format, they will not follow this rule. So we need to resolve the secondary structure into linear form. We use formaldehyde to do that. Formaldehyde makes it linear and also a little bit heat that helps it to denature and RNA will migrate. So once the agarose gel electrophoresis is complete, so we can have the bands of the pattern of where exactly the different fragments of RNA are present. But the question is, agarose gel is fragile, so we cannot use agarose gel for the probing process because for the probing we need to apply different solutions and buffers and little bit weight, and all these things will not be enough for holding all those tensions by this agarose gel. So it is a better idea to transfer the content of gel into paper. That is usually in case of northern blotting was nitrocellulose filter paper. Now in case of northern blotting, we can also use nitrocellulose filter paper, but amino benzyl oxymethyl filter paper are more useful instead of nitrocellulose filter paper because it has more affinity and greater binding affinity towards RNA. Hence, this filter paper are considered as better transfer medium in case of northern blotting. We take this gel and put it in the tank to start the process of imprinting or transferring the content of RNA from agarose gel to the filter paper. And that is when the blotting terms comes in. Blotting is the imprinting of a gel material onto the paper. This blotting technique of northern and southern blotting is very similar because both technique uses the process of capillary action for transferring the content of DNA or RNA from the gel to the membrane filter paper. Now let's have a look at this total blotting setup. We have a buffer as you are seeing in this slide. Then on the top of the buffer, we put sponge. The sponge helps in the capillary activity. And on top of the sponge, we have the gel. And on top of the gel, we put the amino benzoic C-methyl filter paper. And on top of the filter paper, we put some paper towel. And on top of that, we put some weight. So this step is kind of seen in case of southern as well as northern blotting. Now the idea is the buffer will flow based on the capillary activity from bottom towards top. As we apply some weight from top according to the capillary flow, water can flow against the gravitation and it will flow from bottom towards the top. So as the water is moving, it moves first through the sponge, reaches to the gel, it passes through the agarose gel, and as it passes through the gel, it creates a force to all those RNA fragments that are present there. So it will be pushed, and the RNA fragments are now released from agarose gel, and they will migrate towards this filter paper. And once this RNA fragments will go and attach to the filter paper, there is no way those RNA can migrate to other place because filter paper is not permeable to RNA molecules. So the RNA will be stagnant. It will be attached to the filter paper, but the buffer solution can easily pass through the filter paper and it will reach to the paper towel. So this is how the whole process works. Now the next step is we exclude everything. For example, sponge, the gel, the paper towel, and the weight. And we take the membrane out, and now the membrane will have the each imprints of all those RNA fragments that were present in the gel. And now we add probe which is designed to go and bind with the exact targeted RNA. And this probe is a radio labeled. It will emerge light effects. And then we put an extra film over this ABM filter paper, and we will find only one band where the target DNA probe was present on the autoradiogram. And remember that in northern blotting, RNA DNA hybridization works, in which the probe is DNA and it interacts with the targeted RNA. So, this is how the whole northern blotting process works.